So the first one is the Pediatric Immunotherapy Translational Science Network. Subcommittee chairs Dr. Shannons, and then other members are Dr. Martinez and Parada, and the presenters, Dr. Malcolm Smith.
And how's the
Just your forward, backward, that's your laser. And the belly's
okay. Good morning. I'm Malcolm Smith. I'm speaking on behalf of the Pediatric Immunotherapy Implementation Team. Judy Mites and Nita Seibel are the co-chairs of that, and also acknowledge up front our BSA subcommittee with Kevin Shannon, Luis Parada, and Elena Martinez. To begin with background on pediatric immunotherapy, and one of the key points that the pediatric working group of the Blue Ribbon panel made was the distinctive nature of pediatric immunotherapy versus adult immunotherapy. I think you've heard over the last day and over the last months of the focus of adult immuno-oncology research primarily being on agents that boost the immune response to tumor neoantigens. When you look at the agents that are in development, over 20 agents targeting PD-1, PD-L1, other agents, the CTL4 and other agents, all primarily targeted at boosting the immune response to tumor neoantigens. Tens of thousands of patients committed, hundreds of clinical trials, and yet these approaches and these resources that are being spent are likely to have limited applicability for childhood cancers, at least in the way that they're being applied for adult cancers. This is because pediatric cancers generally have low mutation burdens and corresponding low rates of neoantigens resulting from those somatic burdens, somatic mutations. One of the more famous figures in molecular oncology, this is the somatic mutation frequency in a range of cancers. The cancers that respond to the checkpoint inhibitors are over to the right: melanoma, lung cancers, bladder cancer. And when you look at the childhood cancers, they're over on the left. Here, 4 childhood cancers are identified. If we added other childhood cancers that have been sequenced subsequently, they would all cluster on the left as well, with rare exceptions. And so the focus of pediatric immunotherapy research is different. For example, it's on identifying embryonal antigens with low expression in postnatal tissues. It's on antigens that are expressed on cells that are temporarily expendable, like CD19 and CD22 expressing cells for the B lymphoid malignancies. And subsequently, the primary therapeutic translation is engaging the immune mechanisms to target non-mutated epitopes that are differentially expressed in pediatric cancers. So there are examples that these approaches work. A couple antibody-drug conjugates, and we have brentuximab vedotin targets CD30. And this agent is highly active for Hodgkin lymphoma and anaplastic large cell lymphoma in children. We have blinatumomab, a bispecific T-cell engaging antibody that targets CD19, and again, highly effective for children with relapsed refractory ALL and approved by FDA for this indication. And of course, CAR T-cells is another delivery mechanism targeting CD19 and CD22, again, highly effective for pediatric ALL. So there are examples of this approach of targeting non-mutated epitopes working. However, resistance develops, and as well, most these examples are for lymphoid malignancies, and we have myeloid malignancies, and we have pediatric solid tumors for which there's been much less progress. And so the pediatric working group of the Blue Ribbon panel then made these points as background and identified areas of focus that needed attention, and these are the ones that we're incorporating into the Discovery Pediatric Immunotherapy Discovery and Development Network. For example, identification of antigenic epitopes that are uniquely and abundantly expressed on childhood and adolescent cancers, The development of optimized, highly specific binders for these novel pediatric immunotherapy targets, the incorporation of these binders to these special epitopes into novel immunotherapy agents, the identification of mechanisms of resistance to these immune therapy approaches that are pediatric distinctive, and then the development and application of approaches for in vivo preclinical testing of these pediatric immunotherapy approaches using immune-competent pediatric cancer models or humanized mouse models. And so to operationalize these areas of focus, and again, not being prescriptive that these are the only areas of focus, but as examples of the types of research that needs to be done, the proposal is for the Pediatric Immunotherapy Discovery and Development Network. It's envisioned as a collaborative research network with at least planned annual meetings to promote collaboration. Feedback that we got from the BSA subcommittee not only changed the name of the proposal, which I didn't point out, it's different from the one that's in your concept proposal to more specifically describe what we're looking for, but also encouraged us to use a more balanced approach between the allocation of resources to the U54 and UL1 mechanisms. So one part of this is U54 multi-component programs to support collaborative investigator teams addressing two or more relevant areas of focus. For example, one could envision a group of researchers identifying epitopes, another group with expertise in in making binders, and another group with incorporating those into appropriate delivery vehicle, for example, a CAR T-cell. And these teams would work together in a U54 program. We as well have UL1 research programs to support discrete individual or multi-PI projects addressing a relevant area of focus, for example, mechanisms of immune evasion or model development or validation of single targets. We intend to have some NCI core resources to support the network teams and to support advancement of their discoveries to clinical testing in children, and then funds set aside as supplements for collaborations for years 2 to 5 to encourage the different teams in the network to work together and to encourage them to seek collaborators outside of the network when that's appropriate. So what we're proposing, U54 multi-component programs 1 to 2 for 5 years, UL1 research projects, and then collaborative supplements for years 2 to 5. We'll have 2 receipt dates, one for FY18 and one for FY19. And then just to give an idea of how this fits into our overall pediatric immunotherapy research activities at MCI. You know, one way the Cancer Immune Monitoring and Analysis Centers will be established in the coming year. These will provide immune monitoring assays for NCI clinical trial consortia and groups. And one can imagine that if a novel mechanism of immune evasion were identified through the network, that this could then be incorporated into one of the CIMACs and then studied in relevant pediatric immunotherapy clinical trials. And as well, of course, if the network is successful in developing agents, pediatric immunotherapy agents, then these could be transitioned to study in one of our pediatric networks, with the pediatric networks being highlighted in red in this slide. So in terms of markers of success, we will consider our efforts successful if novel antigenic epitopes are discovered, binders are developed for these epitopes. These are incorporated into novel immunotherapy agents. Models for preclinical testing of pediatric immunotherapy agents are developed. Mechanisms of resistance are evaluated and incorporated into clinical testing. New collaborations are established that move pediatric immunotherapy agents forward. And of course, the entry of novel immunotherapy agents into pediatric clinical testing. So this is my presentation. I think Kevin now would be represent the BSA subcommittee.
Yeah, thanks, Malcolm. That was a really excellent summary. I think the sense of the subcommittee was to be very positive about this, but also to try to think about it in terms of added value to the overall efforts in immunotherapy in the NCI and in the Moonshot. As Malcolm said, the original title of this was translation, but when we actually read the RFA, it seemed pretty obvious. It was more pre-translational, building resources, identifying candidate epitopes, testing strategies, building models. And so we encouraged the NCI and Malcolm obviously and his colleagues obviously listened to think about making this less of a U54 and more of a UL1 discovery-oriented mechanism. I think the points Malcolm made are very relevant. They came up in our conversations in the pediatric working group, that because of the low antigenic burden in pediatric cancers and probably some unique antigen epitopes, there probably does need to be an effort to go beyond checkpoint inhibitors, checkpoint blockade in pediatric cancers to look more at cell-based and other biconjugate antibody types of immunotherapies. An important aspect of this is sequencing antigens and detecting antigens in normal developing tissues, because one issue that could come up in these young children is retention of some of these growth associated epitopes and antigens early in life with accompanied toxicity. So not only an effort to sequence the tumors themselves and look for epitopes, but to see which developmental antigens might be expressed. And of course, some of these may come back and be expressed in adult cancer, so there may be some payback or payoff for the adult side of the house if some of this epitope detection is successful. As I say, I think we were very positive about this. We felt that having two different RFA dates was important to allow the NCI to see what came in the first round and the quality, and to have an opportunity for proposals that weren't incisive or compliant exactly to come back and amend their applications, pull different teams together, and also to try to balance out the UL1 and the U54 in the network, and hopefully, as Malcolm said, to interact with other aspects of the cancer immunotherapy that's being done under the NCI Moonshot. So we were very positive and appreciate the sort of responsiveness of Malcolm and his colleagues.
Comments from Elena or Luis?
I have very little to add. I would just again thank the group for being so responsive and amending it in the way it did, which I think brought everything into play that we had been considering. This is an underrepresented area of research, and I think that if you just take the proof of principles from adult immunotherapy with the acknowledgement of the reduced mutational burden of childhood tumors, there are clear avenues of discovery research that just need to be pounded home, and so we're very supportive. Thank you.
So I agree, and I will only add that we had discussions, and to Electra's point about the disparities issues, and for some of the target, even though this is more of a discovery arena, for some of the target cancers that you mentioned, Hispanic children have the highest incidence and mortality, so it will be important to include that in this endeavor.
Kevin, you want to make a motion for us to discuss?
Yeah, I would make a motion that that the board approve this concept, RFA.
Okay. Open for discussion, Liz?
So I think this was wonderful, and it definitely was, the intent was discovery that we really don't understand at all, and kids' immune system is different, their tumors are different. Is there a tissue bank, biobank that could be looked at as well? I didn't see that in there, and I think as you pointed out, the immune checkpoints are going to be different. The pressures within a pediatric tumor to evade the immune response are likely going to be extremely different, as are the T cells that you want to activate. So I think, focusing on the fact that there are biological differences in a young person whose immune system is uneducated to many antigens, and also well, that would also probably lead to different checkpoints.
Right, and so I think that the latter point just goes to the point made about the need for UO1 investigators to really drill down on what are novel pediatric mechanisms of sensitivity and resistance that would,
But you're not going to get it from a mouse model, so you need to have tissue banks to be able to study that.
And so the proposal itself does not include tissue banks per se, but the expectation would be that these would be collaborators that would access COG tissue banks, that would access institutional tissue banks, as needed to get the materials that would be relevant for their proposals.
Just a follow-up on Liz's question. So in your diagram with all the network linking between the activities, are any one of those groups responsible for banking of these tissues?
Well, the Children's Oncology Group, it would be our largest group for banking. You all know that there are 200 institutions throughout the country, and collecting tissue on many of the children that present to their institutions. And so they're a great resource in terms of having tissue available and for these kind of studies.
Other yes.
I just had one question. Malcolm, very nice. I was wondering if you're going to expand to adolescents or young adults, just to encompass that group of individuals that could be more like adults, but also they're kind of the lost group. So I wasn't sure, because that's not in the in your proposal, but I think it's an important group too.
It would certainly, I think the board probably recognizes that when you get into adolescent cancers, that there is this adolescent young adult, that the sarcomas aren't, just a stop at age 18, that Ewing sarcoma is going across the, into the young adult range. And so I think, and so certainly, that those cancers that are adolescent cancers that span the young, the young adult age range would be appropriate for study.
They're often lost in COG, so it's important to expand to that group that could very much benefit from these treatments. Thanks.
Tim?
As it stands right now, the adult and pediatric immuno-oncology incentives are completely separated. Isn't there an opportunity for synergy? Is there some way that could be incentivized in these proposals?
Yeah, and I think they both are envisioned as networks, and so we certainly can. I think it would be one of the jobs of NCI to recognize where the areas of potential collaboration, potential meetings that would be, that would really help, move the field forward, because both groups are attacking the same problem. The point was made, though, that a lot of the checkpoint blockade just won't be as relevant, and so to the extent that a whole swath of the adult is focusing in that area, that may not be relevant, but there will be other areas that will be relevant, and we should pay attention to those.
So, Tim, I think what we are hoping is that many of these initiatives are going to interact. So there should be areas of intersection among the immuno-oncology groups, but also with the Human Tumor Atlas and with the technology development groups. So the goal will be actually to promote those interactions.
Charles?
I have more of a high-level question for strategy at the NCI level about U54 versus U01. Just looking, it's not really specific to this particular proposal. Just looking at this, it's roughly $20 million for the U54 for 2 proposals, and then the U01 total budget over the 5 years is $4 million. So if I understand it, for at least this particular one, we need a lot of new innovative ideas probably here more than adult. So how do you decide this balance?
I think just one point, as a result of the BSA subcommittee, that what will actually end up going forward is a more balanced allocation with, approximately equal funds. So it'll be more U01s and correspondingly less U54s. So for this proposal, it's less U54s, but I'll let others respond to the more general question.
Charles, I think you're asking a really important question, and we wrestle with allocation all the time. What I hope will happen is that we will be able to make awards based on the overall quality of the applications, and in some instances to make more awards than the set aside.
Okay, other, yes.
Yeah, can I just put in a plug for the U54 with the funds inside the U54 to spin out additional collaborative projects down the road? In our past life with the U54 and energetics, that was one of the really unique features of having the U54s, and really the potential to be responsive over the life, and that feels like a real strength here in this proposal.
Okay, any other comments? I guess we heard the discussion, so I'd like to see all those who are in favor of approving. Any against? Abstention? Okay, pass.